Hello, everybody, and welcome back to the lecture on Chapter 10 on controlling microbial growth in the body. The last slide for part one, we had gone over these four mechanisms of action that these antimicrobial drugs have. In this part of the recording, we're going to hit the inhibition of cell walls, both in bacteria and in fungi, and we'll also do disruption of plasma membranes, both in bacteria and in fungi. And then I'll save number 3 here, inhibition of protein synthesis and inhibition of metabolic pathways, for the next part of the lecture. All right, so first let's do inhibition of cell walls. And that's a kind of a general mechanism. Both bacteria and fungi have cell walls, but they're different structures. So a drug that's going to attack a cell wall or affect a cell wall in bacteria is not going to be effective against fungi, and vice versa. Something that's going to work in fungi is not likely to work in bacteria, again, because of the so-called selective toxicity. It's going to work on whatever structures are targeted. So bacterial cell wall, let me remind you, this is largely NAG and NAM subunits. This is the so-called peptidoglycan layer. And then between the NAM units, the NAM is the one that's represented in the lighter color. Between those NAM subunits, there are these cross bridges that have to be formed by particular enzymes that link those things together with covalent bonds. So as the new cell wall is being formed for like the next generation, and new subunits are coming in, the NAM subunits are tied into toward each other. So most of the antibiotic agents that work on the cell wall actually work right here, is preventing that cross linkage of the NAM subunits. The most prominent kind of drug in this group, they're referred to as the beta-lactams, and I do need you to know that. As in nursing, although we will not be prescribing the medications ourselves, we are the ones that administer the drugs. Let me just tell you what happens in the clinic. You'll find out that a patient has a penicillin allergy, which is a beta-lactam, and the patient is prescribed amoxicillin or methicillin. And it's not exactly penicillin, but it's close enough related in structure that as a nurse, you might need to observe the first dose of the medication to see if the patient's going to have an allergic reaction. And that's why it's helpful to know which medications are, sharing this beta-lactam structure. Okay, so the functional groups are beta-lactam rings, and what that means is chemically the bit that does the work is what's shown here. This is penicillin G. This is the thing that comes from penicillium. This shown in green, this is the so-called beta-lactam ring. It's some carbons and then nitrogen in one corner, carbon with a double bonded oxygen. Doesn't matter. Point is, this green thing, that's what interferes with the enzymes that bind the NAM units together. So that's the so-called beta-lactam ring, and you can see it in other groups that are other drugs that are also beta-lactams. Methicillin, for example, and cephalosporins are also beta-lactams. Okay, there you go. You got all the names. It's that beta-lactam, that's what binds to that enzyme and is going to interfere with the NAM units connecting to each other. Now, why does that matter? Well, if you expose the bacteria to all these beta-lactams, when the bacterium is trying to reproduce and make new cell wall for the daughter cells, then these NAM units these beta-lactams getting in between the NAM units makes it impossible for the covalent bond to form. And so this thing later has this weakened cell wall, and when it's exposed to just normal osmotic pressures, the thing just breaks open. Hooray! We can fight off bacterial infections like this. Fantastic. So you know that there are many more antibiotics than just penicillin. So why would we ever need anything else? Well, it turns out that evolution happens and we get the development of resistance. In other words, this is what's referred to sometimes as an evolutionary arms race, where one species evolves one mechanism and then the species that it's fighting off against develops through random mutation, develops some way that it beats this defense. And so one of the ways that some antibiotics have evolved is something known as a beta-lactamase. It's an enzyme that actually snips that little green square chemically and makes it non-functional. So where you have, here's that beautiful beta-lactam ring, and the bacteria comes back with, oh yeah, watch this, and just snips this thing. So this is no longer going to get in the way of the enzyme that we were trying to get in the way of. So that's one example of bacterial resistance to antibiotics, and we will talk about it later in this chapter. But that's it for today. Well, that's not it. I'm going to tell you, we didn't give up. This is an evolutionary arms race, right? So then we develop a drug that fights the beta-lactamases. We call them beta-lactamase inhibitors. I think of it this way. Like, here's the beta-lactam that's going to do the damage. And then the bacteria comes back to us with little scissors where they're going to snip the little beta-lactam, and then we come back with a rock and we crush the scissors. So the drug that crushes the scissors can do nothing by itself, but if it's given at the same time that we're giving a beta-lactam, then yes, we can fight off the bacterial resistance to the antibiotic. Just genius. Clavulanate is the scientific name. The name that's given in the clinic, you may have heard of it as Augmentin. So more info on resistance later in the chapter. Let's continue with what we can do with, fighting with this the cell wall, again, because we can focus on that cell wall because humans don't have the cell wall. So other drugs that also focus on the cell wall are semi-synthetic derivatives of beta-lactam. So that we had the penicillin originally, and then vancomycin, you may have heard of. This interferes with specific links between the NAM units in the gram-positive bacteria. Bacitracin, which you may have heard of. This is often used as a topical medication. This blocks transport of NAG and NAM units from the cytoplasm. So it has a different mechanism of action, but it's still working, like focusing on that cell wall that the bacteria have that we don't have. Isoniazid and ethambutol. These are specific, they're focused on cell wall, interfering with the cell wall, but it's specifically in gram-negative, it's specifically in mycobacterial species. Things like tuberculosis is when you'll see isoniazid and ethambutol being administered. Now, this is an important point. All of these drugs interfere with the formation of the cell wall. It doesn't do a thing about existing cell wall. If those subunits, the NAM subunits got bound together, this isn't going to break that. It can only interfere in the formation of those covalent bonds. So it works when the bacteria is actively reproducing. Here's the tricky thing. Some of the bacteria reproduce very, very slowly. So it's effective only on growing cells. So the dormant cells are unaffected. And these slow growing cells, like tuberculosis, is notorious for being slow. its generation time, if memory serves, it's something like a couple of generations in a day, which I know sounds really fast, but it's ridiculously slow for bacteria. So those drugs, they have to be administered for months, in some cases years, to get a bacterial infection like tuberculosis, to get that completely cleared. And that's one of the reasons why treatment of tuberculosis is so challenging, because you have to keep at it and keep at it and keep at it. And that's just a really challenging thing to do clinically. Now let's move on to inhibition of the cell walls, but this time, instead of the bacterial cell walls, we're talking about fungal cell walls. Those are a little bit different. I'm not going to talk about this for very long. The fungal cell walls are composed of a variety of polysaccharides that are not in mammalian cells, so we can use those as the targets. Medications called echinocandins, those inhibit the enzymes that synthesize glucan, the polysaccharide. And just like what we talked about with the bacterial drugs, or the drugs against bacteria, it causes the cell wall to fail. All right, so that was attacking the cell wall, first for bacteria. I gave you a whole long list of drugs, most notably the beta lactams. And then we shifted focus to the cell walls of fungal cells. Now we're going to shift focus we're going to go in a little bit from the cell wall down into the plasma membrane. This is sometimes referred to as the cytoplasmic membrane, and sometimes called the cell membrane, plasma membrane, cytoplasmic membrane, all mean the same thing. It's the membrane part that they have in common that's different from the cell wall. First, let's have a look at doing this in bacteria. The drug that's in this category is known as polymyxins. These disrupt the cytoplasmic membrane of gram-negative bacteria. The trouble is, these are really toxic to human kidneys. And so it's a no-go for using it systemically. But if the infection is on the outside, if it's a skin infection with a gram-negative bacterium, this might be a reasonable drug to use, because it can be used topically, but cannot be given systemically or orally, intravenously. That's a no-go, because it'll damage the kidneys. Okay. And then, so not a lot of information there, because there's not a lot of drugs in that class. And now let's take a look at disruption of the cytoplasmic membrane of fungi. And we have a few drugs in this category, but the thing to really understand about them is that the drugs that fall into this category, they work by, one of the ways that they work is by creating a channel, an opening in the cytoplasmic membrane, damaging its integrity. And so the cell is not able to maintain a difference between the inside of the cell and the outside of the cell, which you know is kind of critically important. The drug name is a little bit complicated, amphotericin B. How it works is that it attaches to a structure that's in the cell membranes. Remember, what all the cell membranes have in common is this phospholipid bilayer. So if you attack the phospholipid bilayer in and of itself, like, you're going to damage human cells also. So that's a no-go. So we had to find a target that's different between human plasma membranes and fungal plasma membranes. And one of the things, if you remember, the plasma membrane has those phospholipid bilayers, the little lipid tails go in towards each other, and the heads are out towards the water that's inside the cell and outside the cell. Okay, another thing that's embedded in our cells are cholesterol molecules. That's the sterol, it's a steroid molecule that's in there. Fungi don't have cholesterol, but they do have a sterol, and it's called ergosterol. So ergosterol, like cholesterol, there's the sterol. So humans are a little bit susceptible. This medication does damage human cells a little bit, because cholesterol is similar to ergosterol, but let me show you how it's supposed to work. So here's that phospholipid bilayer cartoon, the phosphate heads on the outside of the cell and on the inside of the cell, and the lipid tails in towards each other. This is what I sometimes refer to as the Oreo cookie layer. Then here's the sterol embedded. In us, this would be cholesterol. In fungi, it's ergosterol. And then the drug, amphotericin B, works like this. It connects to the ergosterol and essentially creates a great big hole by bringing a bunch of these ergosterols together and making an opening. Just genius. So that's amphotericin B. There are some other medications that also target the ergosterol structure, but in a slightly different way. Oh, whoops, sorry. Let me make this point too. We've mentioned this before. It has to do with the selective toxicity. Since bacteria don't have sterols in their cell membrane, this medication is completely worthless against bacteria. So it won't treat a bacterial infection. Okay, and then finally, let's get to the last medications in this group, azoles like fluconazole. Some of you may have heard of it called diflucan. Allylamines, these inhibit the formation, the synthesis of ergosterol. And since we weren't making ergosterol anyway, this is not going to affect our cells. These are the most common antifungal agents. I covered them later because you kind of needed to see what ergosterol was first before you could understand why would this be a problem. But if you can't make this at all, if the fungi cannot make the ergosterol at all, that affects the ability of the cell membrane to have any kind of fluidity to it. And so azoles are quite effective against fungi without having an impact on the human cells. All right, so that is the end of part 2. I will see you over in part 3, where we'll cover those last two mechanisms of action of antibiotic drugs or antimicrobial drugs. See you over there.